So in this second session, we're going to start with a talk from Jared Simpson. Jared's a principal investigator at the Ontario Institute for Cancer Research and also has an appointment in the Department of Computer Science at the University of Toronto. And Jared has been a collaborator with ARTIC since the start. He is the developer of the tool Nanopolish that we've already spoken about in the first session, that's used in the ARTIC bioinformatics pipeline, a really important tool for analyzing nanopore data. And Jared, like so many of us, has been working on coronavirus genomics intensely for the last year and a half. As part of the Canadian COVID-19 response and their consortium project in Canada, Cancogen. And one of his roles there has been as the kind of curator and guardian of data quality at Canada, and has a lot of experience with staring at coronavirus genomics data and working out what the heck is going on with it. Something I also spend quite a lot of time doing. So we're hoping to get some good insights from Jared now about how he tackles analyzing SARS-CoV-2 sequencing data and the package that he's developed. So thanks very much for joining us early in the morning, Jared, and over to you for your presentation.
Great, thanks Nick for the introduction. Really happy to be here and share what we've learned. So as Nick mentioned, I'm going to give sort of the perspective of what we're doing for quality control here in Canada for Cancogen, and focus mainly on this toolkit that we've been developing in my group, mainly led by Richard DeBorgia, who's on this call here today, called ncov-tools, which is the package we use to run on all sequencing data generated here in Canada to ensure that it's high quality before releasing to the public community. So I'm going to start by giving an overview of Cancogen. So as Nick mentioned, it's our national SARS-CoV-2 sequencing project. It's a distributed project. We don't have a single centralized sequencing facility that does all the coronavirus genome sequencing in Canada. Essentially, every province has one or sometimes multiple sequencing sites, typically led by the public health labs within each province. So it can be seen as a large distributed sequencing project, which is a collaboration between public health, universities, and research institutes like my institute at the Ontario Institute for Cancer Research. So being such a large and distributed project, we needed very strong coordination and to ensure everybody was using the same sequencing protocols, the same bioinformatics approaches, and using the same criteria for determining whether a sequenced genome was sufficiently high quality to use for downstream analysis. And that's where the quality control working group came in. We struck this working group very early on in the project, well over a year ago now. They would decide on exactly which quality standards we would use, and then develop the software package ncov-tools to be able to be run to perform those quality checks. So the quality control working group is quite large. I'm not going to read all of the members, but this will serve as my acknowledgement slide. I just want to highlight Richard DeBorgia, who I mentioned is on the call here. Finley McGuire, Hector Galvez, and Natalie Knox all made key contributions to developing both the quality standards and testing and implementing them in this software package. So now I'm going to get into talking a little bit about genome quality. So we can think about quality in different terms. At the most basic level, we might think about checking just whether the genome was successfully sequenced. So almost always, at least in Cancogen, we're starting from clinical specimens, which may vary in the amount of virus that's actually present in the sample. And the amount of virus that you have in your sample determines how well it's going to be sequenced. If there's very little virus, we may not get complete coverage of the genome. If there's a lot of virus, it's going to amplify well, and we're going to get better coverage and hopefully be able to recover a complete genome. So we're going to want to develop a way of quantifying exactly how well each sample sequenced, and then using that as one of our primary methods for QC. Assuming we've then got a sequenced virus, we might want to check whether the genome sequence that we've determined, the consensus sequence, is accurate. Again, when we have low virus abundance in our samples, we might have things like amplification artifacts that can cause miscalls in the consensus, or if the genome just isn't covered well enough, that might lead to other types of consensus errors. Or you might just have bioinformatics problems where the software we used to determine the consensus sequence may have made an error. So we want to develop methods for trying to quantify whether our sequence is accurate enough to use in any sort of subsequent analysis. And finally, we want to check for technical issues that went on during the sequencing run. So we want to check for contamination. All of the sequencing protocols are highly multiplexed. You sequence dozens, if not hundreds of samples simultaneously, typically in plate format. And you can have cross-contamination between different wells of your plate. And there's, as Josh told you in the morning session, there's typically very many cycles of amplification that go on. That means you may get a lot of subsequent DNA that can go on and contaminate other samples in your well. So we want to develop methods for detecting whether any samples have been contaminated and flagging any problematic runs using those criteria. So I'm going to start by talking about the most basic QC, which is just, was our genome successfully sequenced? Now, this is a plot from ncov-tools. I'm going to be showing a lot of plots from ncov-tools. I think all of the figures I show today will be generated by the tool package, and you'll be able to generate them on your own data if you use this software. So this is just a basic coverage plot, which shows the sequencing depth for every position of the coronavirus reference genome. So from position 0 over to position 30,000. And this is logarithmically scaled. So here we have depth of about 1,000. And we can see that this sample sequenced very, very well. This coverage is fairly uniform at 1000x. And that should present no problems for the consensus programs to determine what the sequence of the genome is. Now, these plots are typically annotated with a little bit of metadata, like the qPCR CT values. This sample had a CT of 16, so we would expect there to be sufficient viral material to amplify and sequence, and that's reflected in our coverage profile, which looks really quite healthy here. If we go to a sample with a higher CT of 25.3, we see that there's starting to be some problems with our coverage profile. We can see some dropouts where the coverage drops down below 10x or even down to 0 here, here, here. And we can see some other areas where some of the amplicons didn't perform very well, like here, 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 and maybe here. So we might not be able to recover, well, we definitely won't be able to recover a complete genome in this case, but we may be able to cover a genome that's complete enough to use in downstream analysis. And we want to be able to quantify exactly how much of the genome we've recovered, and we're going to come to that in just a minute. If we go to a sample to be considered very high CT, CT34, we see that the coverage profile looks very, very poor here, with the majority of the genome being not covered by sufficient reads for calling a consensus sequence, and we'd be unlikely to get a high enough quality genome to perform any sort of analysis on later on. So the way that we quantify whether we've recovered enough of the genome for analysis is by calculating what we call genome completeness. Now, the bioinformatics pipelines, like the Nextflow pipeline that Matt Bull presented earlier, are typically reference-based in that they're going to take the sequencing reads, map them to the reference genome, then decide whether there's enough coverage to call a consensus base at each position of the genome. For Illumina data, that's typically about 10x coverage is required to call a consensus base. For nanopore data, it's more like 20x coverage. And any positions that don't meet those thresholds will be masked in the consensus sequence with ends. So ends are IUPAC ambiguity code. which indicate that there's not enough, there's no information about what the true base is. So it could be any of the four possibilities, A, C, G, or T. So we define genome completeness as the portion of non-N bases in the genome sequence. So here's just two toy examples, sample A and sample B. Sample A has 3 N's, 7 ACGT bases from a genome size of 10. So we would say that the genome completeness is 7 divided by 10, or 0.7, or 70%. In contrast, sample B only has 1 ambiguous base, and we would calculate the genome completeness as being 90%. So we can directly go from a coverage profile that I just showed you to a consensus sequence where the positions that had insufficient coverage are marked with N's, and then we can calculate our genome completeness from that consensus sequence. Now, the reason we use this as a QC criteria is that incomplete genomes are much more difficult to analyze. You may not be able to reliably assign a lineage using a tool like Pangolin, which you'll hear about tomorrow, or it might just be difficult to construct a multiple sequence alignment to build a phylogenetic tree if your genomes are highly incomplete. So many projects, I think probably all projects, use genome completeness as one of the primary criteria for determining whether a sample is good enough to analyze, and more importantly, also whether it can be released publicly into large sequence collections like GISAID or other data repositories. And for Cancogen, we require that the genomes must be at least 90% complete for them to be publicly released into a data repository. Now, there's a very well-known and expected relationship between qPCR-CT values and genome completeness. And here I'm plotting many samples that have been sequenced at my institute, OICR, where each point is a sample plotted based on its qPCR-CT value and this genome completeness that we calculate from the consensus sequence. As we can see that when qPCR-CT is less than about 30, we typically get very complete genomes, over 75%, and very often nearly 100% complete, where we've recovered the entire genome. But after about CT30, we start to get a drop off where we have much more of the genome being not covered by reads. So for this reason, many projects have made the choice that they're not going to try to sequence samples beyond a certain CT threshold. In Cancogen, we don't sequence beyond any samples that has CT above about 30. And that's really just to save cost, as beyond about CT30, you are unlikely to get a fully recovered genome. So you'd just be spending a lot of money to sequence things that are unlikely to be successful. Now, another way of looking at coverage is by this heat map that's generated by ncov-tools. So in this plot, we're summarizing the coverage of all samples on a run for every amplicon. So here, each amplicon is a column, and each sample is a row. And each square is filled in by the depth of sequencing of that amplicon, where blue color represents very high sequencing depth, and red color represents very low sequencing depth. So immediately we can start to see patterns in this plot. We can see that these three samples, they have a lot of red boxes or red shaded boxes, which indicates very low sequencing depth. Now I know, because I prepared the slide, that these were the negative controls on this sample, so we'd expect them to have quite low coverage. We can see that there's some real samples like these two, this one, and a few in this block here that didn't sequence very well. But we can also see patterns in the amplicons as well. For example, this column all show amplicon 64, which has lower coverage than the other amplicons on the sequencing run. Also, amplicon 70 and 74 had consistently lower coverage. So this shows us that these are the three weakest amplicons on there, and it might lead you to believe or think that you might want to try to tweak your PCR conditions to try to improve these weaker amplicons. So this just gives you a quick one image summary of how well each amplicon performed across all of your samples on your sequencing run. Now I'm going to move on to talking about genome sequence accuracy now. So assuming we've sequenced this complete or nearly complete genome, we want to determine whether the consensus sequence that the bioinformatics pipeline has determined is accurate enough to use downstream. Now, as I mentioned, ARCTIC data is analyzed by comparing reads to the coronavirus reference genome. So in this little illustration, the reference genome is shown in blue. All of our reads for our sequence sample is shown in black. And our consensus sequence that the bioinformatics pipeline determines is shown in red. And I'm highlighting one position here where there's a T in the reference, and all of the evidence in the reads for this sample also show a T. So we would put a T in our output consensus sequence, which is what we've done here. So this position has strong evidence that the sample we've sequenced matches the reference allele at this position. If we look at a different example, here we have a T in the reference, all the reads aligned to it show a C, and we would put a C into our consensus sequence. And we would call this either a variant or a mutation at this position. Now, ncov-tools generates a plot that summarizes the variation present in your sequenced samples, which I'm showing here. And this is work that was inspired by some plots of Mads Albertsen of the Danish Sequencing Consortium generated. Now, in this plot, all the samples are ordered by a phylogenetic tree that's built by MAFT and IQ-tree. And then we depict with these colored boxes on this heat map-like diagram all of the variation present within every sample on your sequencing run. So whenever you have a colored box, it means that a sample mismatched the reference genome, had a variant or a mutation at this position. So these purple boxes here that I'm just showing with my mouse pointer all show that every sample in our sequencing run had a T mismatch with respect to the reference genome at this position. Now we can start to see groupings or clusters of samples on this plot. All of these samples here seem to show the same pattern of variation, and that's consistent with this little subtree on our phylogenetic tree. All of these samples here are clustered together as well, as they share this 3 base pair substitution at the 3' end of the genome. And there's a little subcluster here of samples that share these 5 substitution variants at these positions of the genome. So this plot just gives you a very quick overview of the variation that's present in your genome, and you can start to look to see whether the patterns of variation are consistent with the groupings that you expect in your samples. Now, we also need automated ways of determining whether our samples are high quality enough. And one of the automated checks that we do is looking for ambiguous or mixed bases in our consensus genomes. Now I'm going back to this illustration of reads piled up against a reference genome. Here we have a reference T, but instead of all of the reads in the sample showing a single base, we have a mixture of evidence where some reads show a T and some reads show a C. Now, for when you're processing Illumina data, the pipelines will annotate these positions using what we call IUPAC ambiguity codes. These are extended nucleotide representations which flag positions that might have more than one possibility. So in this example, we put the IUPAC ambiguity code Y into our consensus sequence, as that's the ambiguity code that indicates a C or a T at that position. So essentially what's happening here is that the bioinformatics pipeline, the consensus caller, has determined that it can't decide between a C or a T at this position. So it's put a Y into the consensus to indicate that it's ambiguous. And we call these ambiguous bases or mixed bases. There's a lot of different reasons this can happen. There can be true biological reasons for having mixed bases, like intra-host variation, or there can be sequencing artifacts. If you start from very little viral material, you may get early PCR amplification artifacts that get into the reads, or you might have errors during reverse transcription that cause there to be multiple populations of reads at different positions of the genome. Now in CanCoGen, we track the number of ambiguous bases present in the consensus genomes. And again, I'm plotting the number of ambiguous bases seen in our consensus genomes as a function of our qPCR-CT values. And again, we see the same pattern where less than CT30, the genomes tend to have very few mixed or ambiguous positions, but beyond CT30, the number of mixed and ambiguous bases goes up quite sharply. So this indicates, to me at least, that it's likely driven by sequencing low amounts of virus and probably something like an amplification artifact or an RT artifact rather than true biology. So in our analysis pipeline, or sorry, in our QC pipeline, we flag any samples that have 5 or more ambiguous or mixed positions. And these samples show up in the SNP plot that I showed you previously as, rather than colored boxes, black boxes here. So whenever you see a black box in the SNP plot, that indicates that the position was ambiguous in those samples. So this is just zooming in on one of the little groups of samples that we were looking at, and we see that this sample here, third from the top, has 5 mixed positions. So it would be flagged as a QC fail by our bioinformatics pipeline. Sometimes you want to rescue the samples, so you might look at the CT values and see that, oh, if it's a CT32 and it has a lot of mixed or ambiguous position, it's probably some sort of amplification artifact. So you'd then discard that sample or maybe try to rerun it if it was really important to do so. Conversely, if it had low CT, you may go in, look at the evidence of the read level and try to see whether it might be intra-host variation rather than some amplification artifact. Now, a second type of consensus QC that we perform is looking for frameshift mutations. So this is moving away from looking for substitution style mutations to insertions and deletions. Now, the coronavirus genome is very dense with protein coding sequences, and we'd expect very few indels to be introducing frame shifting mutations. So having length that isn't a factor of 3. So we run every consensus genome through a variant effect predictor called either SNPF or BCF-tools to look for any proteins that have a frame shifting mutation in them, like this one does here. If the sample has a frame shifting mutation, we flag it for manual review, as if you try to submit a sample that has a frame shift to a database like GISAID, it will automatically reject it. You can recover those samples if you tell them you've manually reviewed the consensus sequence and think that it's okay, but it's usually an indicator of a lower quality genome, particularly again when the CT value is very high. And we see the same effect where as CT increases, the genomes accumulate more, tend to accumulate frame shifts more readily. So again, that's probably an amplification artifact. So we flag any samples with a frame shift for that reason. All right, finally, I'm going to talk a little bit about contamination. So the last question we might want to answer about our sequencing run is whether it was contaminated or not. So the gold standard way of assessing contamination is by running negative control samples in your sequencing run. So typically these are just water blanks where you carry some water, no real clinical sample, no real virus, all the way through the sequencing process from RT to PCR to sequencing and bioinformatics analysis. And in our tool package in ncov-tools, samples can be flagged as negative controls, and their coverage profiles will be specially checked to make sure that there's no reads that may indicate that there was viral material or contaminating amplicons within that sample. So here is what a very good negative control looks like. Unlike the previous samples where we had 1000x coverage, we have very little coverage, only a handful of reads mapped to the coronavirus genome for this negative control. These might just be primer dimers, so we don't worry about these little reads mapping here. What we're looking for is that we don't want high depth of coverage across the entire genome. Here's what a problematic negative control looks like when you have contaminating amplicons present. So here, unlike the previous plot, we have these stacks of coverage at certain regions of the genome. These correspond to amplicons that somehow work their way into the negative control, and they've been sequenced to a depth of over 5,000x coverage. Now, these samples, this sequencing run would be immediately flagged by ncov-tools as having 5 amplicons contaminating one of the negative control. The recommendation would then be either to discard the run and resequence it, or if you prefer, you may want to just mask all of the real samples with ends in the regions that are indicated as being contaminated with these amplicons. Typically in CanCogen, if there's 2 or fewer amplicons in the negative control, we would try to mask them. If there's more than 2, we would discard the run and resequence it. Here is a different type of contamination. Unlike the previous one where we had contaminating amplicons, here we see low-level coverage across a wide range of the coronavirus reference genome. The coverage is fluctuating between about 5 and 10x, but it's widespread across the entire background of the reference sequence. So here the recommendation would probably be to discard the run. If you really want to try to rescue it, you can go in and change the coverage thresholds in the bioinformatics pipeline. So instead of requiring 10x coverage to generate a consensus base, you might increase that threshold to 100x or even 500x to try to recover that. But it would be safer just to discard the run and resequence it. Now, the final check that we perform is looking for individual contamination of single samples on the run. So rather than widespread issues like contaminating amplicons that might affect the entire plate, this check looks at whether individual samples appear to be contaminated. And the way that this is performed is looking at the mixed positions within a sample, like sample C here, to see whether it can be explained by mixing together reads from 2 other samples on the run. So for example, sample B and sample A. Now, if we focus on this mixed position here, we see that sample A had a SNP at this position depicted by the purple box, and sample B had the reference allele depicted by this white box. Now, if you mix together reads from sample B and sample A, you would get a mixed position in sample C here. And the same pattern holds for the other 4 mixed positions in this sample. So this pattern of mixed bases can be perfectly explained by mixing together reads from sample B and sample A. So we have a bioinformatics script that will do this sort of analysis. It's called mixturecheck.py, which will analyze a consensus genome and the coverage profile and allele frequencies to determine whether the pattern variation can be explained by having mixed 2 samples together. This can be simple pipetting errors, like just pipetting 2 samples into the same well, or other things like splash over between adjacent wells on your PCR plates. So typically when you find this mixture pattern, the sample will be flagged and you would discard that one and not proceed with downstream analysis and not release it publicly. Okay, just to summarize the types of QC that ncov-tools performs, it will perform checks across the entire sequencing run. For example, whether there's any amplicons detected in the negative control, checking whether there's no coverage or low coverage across the entire region of the negative control, and check whether no positions are ambiguous in more than 3 samples. I didn't talk about this one, but this is another way that we try to detect contamination by amplicons across real samples. It will then perform a lot of sample checks on individual samples. It will check whether completeness is at least 90%. It will check that you have fewer than 5 ambiguous bases in the sample. It'll check that whether it has any frame shifting mutations and whether there's any evidence that it's mixed with any other sample on the sequencing run. Now that takes me to the last few slides, which is just briefly talking about the software, which you'll have a chance to try in the homework session as well. So as mentioned, ncov-tools is our software package that automates all of these quality checks and automates generating all these coverage plots and SNP plots and the heat maps that I've shown today. It's implemented as a snake-make pipeline and a collection of Python scripts, but the snake-make pipeline will drive it and take in as input the Arctic pipeline results in a configuration file, and then you run it with just a single command where you point snake-make to the master workflow script, give it a target of all, and it will generate the entire QC report for your sequencing run. So essentially what you have to pass in is a analysis run directory that may have come from the Nextflow pipeline that Matt Bull talked about. Within that directory is the BAM files and consensus files for every sample in your sequencing run. You then also pass it some resources files like the Arctic reference genome, the Arctic version of the coronavirus reference genome, I should say, the bed file that's describes the amplicon scheme, and then you just link everything together, telling it where you can find it in the configuration file, and then run this command to build everything. Once it's finished running, you get a report, a single PDF report that looks like this. It will plot the variation present within your sequence samples, perform the mixture checks and ambiguity checks. It will assess the coverage in your negative control and display whether each negative control passed or failed QC in this little table. If the negative control failed QC because of contaminating amplicons, they'll be listed here in this part of the table. And then in the final part of the report, it will list the per sample QC metrics and any QC flags that those individual samples failed. For example, this sample here was flagged with incomplete genome, as the genome completeness fell below the threshold that we would use for downstream analysis. So this report, this PDF report, gives you a nice transferable way of indicating the quality of each sequencing run that you can then share with the lab or anyone interested in the quality of the run. All right, I'm going to finish here with just some links. So our software is all freely available on GitHub. It's probably easiest to install it with Conda. If you just do Conda install ncov-tools, it will install the entire package. Richard's written some very nice documentation for how to configure the various features of ncov-tools and what all of the outputs mean. You can read that on the wiki here. And we've developed a tutorial where you can download some public data, run it through the Nextflow pipeline, and then run it through ncov-tools to generate a QC report that I just showed you on the previous slide. And I just want to thank again all the members of the QC working group and the wider coronavirus genomics community for the discussions around QC standards and methods that helped us develop this package. And again, this was part of CanCoGen, Canada's National Sequencing Project, which is funded in part by Genome Canada. So thank you all for listening. I'll be happy to take some questions. I can see some in the chat here.
Brilliant. Thank you very much, Jared. It's an incredibly clear presentation. And I do think that this is a really important topic at the moment, and a really useful tool set as well. And it reminds me that I should use it more often, and I don't, but I should. Okay, so as you say, we have got some really good questions. Thank you everyone for your engagement in the chat. Please do post any more questions that occur to you. So why don't we take those as they come. The first question is from Miguel, who's picked up on the fact that typically you will not call bases with a coverage depth of less than 20. Could this be adjusted? You know, if you were pretty confident that you know, this is not contaminated, could you go to a lower threshold than 20?
Yeah, it's a good question. I guess we should handle that differently for nanopore and Illumina data. You know, the threshold is 20 for nanopore data. You know, as a developer of nanopolish, I think 20 is probably the lower limit that I'm comfortable with. A lot of nanopolish is based on using the signal data rather than the raw base called reads. And to be able to properly calibrate its models requires quite a lot of data. And I think that if you fell below 20, you'd probably go into a region where nanopolish's models weren't as comfortable with the data as if you're above 20. So I would probably say for the nanopore data, at least to stick with the 20 threshold. For Illumina data, we use a coverage threshold of 10x. That's already pretty low, and I probably wouldn't be very comfortable changing that either. So I'd probably stick with those two coverage thresholds.
Yeah, I mean, and I think even 20x is like on the lower boundary for nanopore data. But I mean, just to follow that up, some centers, for example, will be sequencing to incredibly high depth, which is quite easy to do on Illumina. Like some centers will use a NovaSeq run for 384 samples, in which case they can be routinely producing 10,000x coverage, even higher than that across a genome. In those situations, what do you do? Because even a very low rate of contamination could potentially be an issue if you are accepting a base call at 10x, but your average coverage is 110 or 100,000x. Is there a scope for not thinking about it in terms of absolute coverage, but actually more like, like Josh was talking about, the coefficient of variation and things like that?
Yeah, we've had some discussion recently about that, because this contamination mode that I showed a few slides ago, where you have this low level background coverage of about 5x. You can see that quite often when you do these super deep sequencing runs, like on a NovaSeq or even a NextSeq with like the higher output kit. So we have thought a little bit about whether we need to make that flexible. So if you do sequence of 50,000, maybe 10x coverage isn't the right threshold because things can leak through. I think it's quite hard to formalize. It's nice just to have a very simple threshold that is interpretable. But yeah, I think there is probably scope in making that threshold based on your overall coverage. But we haven't explored that in detail yet.
Yeah, and I think you quite nicely pointed out the need to actually go and look at the data sets. So, a threshold is not so as important as actually investigating runs, particularly when you're starting out, when you're getting your process established, really looking and validating, are you getting back the results you expect, running positive controls, running known samples, and really looking at the output of ncov-tools to understand that. It's really important.
Yeah, just like something I wanted to highlight, but I think I only mentioned in passing, is that there's sort of two philosophies in QC, whether you make hard pass-fail decisions or whether you're flagging problematic problems that should be investigated. In ncov-tools and CanCogen, we sort of lean more towards the latter, where we flag things that then should be investigated rather than making hard pass-fail distinctions. Some of our QC criteria is hard to pass-fail, but a lot of the things I mentioned is more flagging that should be looked at to get some comfort with the data.
Yeah, definitely agree. Okay, so let's carry on with the questions. So Kurt is asking, and this has kind of come up a few times, how easy is it to spot low quality genomes, in the public databases, or, data that's being shared by collaborating centers, and do you have do many low quality genomes actually end up in databases like GISAID?
They do. The most basic QC, the genome completeness, you can calculate on your own from any sample you download from GISAID. You can calculate genome completeness very, very simply. There's a lot of tools out there to do it. If you know a bit of Python, it's very straightforward to do. So that's one QC you can do right away on all public data. Other QC that we were talking about is more difficult. You can look for ambiguous or mixed bases, but that obviously depends on the group that's submitting the data, having used a pipeline, they would introduce IUPAC ambiguity codes. But often you're sort of, the bioinformatics tools and even a sequencing protocol isn't well annotated in the metadata, so you might not have that information of how the consensus sequence was exactly generated. So it definitely can be very difficult to do the more sophisticated QC. I think this is a good chance to sort of make the argument for uploading the raw reads somewhere. I know ncov-qc has been leading the way in uploading the raw sequencing data so that you could reanalyze it and do your own quality control checks. If you're concerned or if you think there may be a quality problem with some genomes that you're trying to look at. We're starting to do that in Cancogen, but we're definitely behind in sharing the raw readings, but we have the intention to. So yeah, the short answer is that you can do some quality checks on public data, but to do a really robust job of quality control, you really would need access to the raw data. And I definitely think there are low quality genomes and just said, both in terms of completeness, but also things like contamination or having like various artifacts that made it through into the public submission.
Yeah, I agree. And that kind of Marina's question kind of follows on from that, which is what do you think about the masking strategy presented by Nicola Di Maio? And she's linked to the virological post about that masking strategy. Maybe you could just describe what that masking strategy is first, and then just say what your thoughts are about that.
Sure. So early on, and I think they've been updating it throughout the pandemic here. Nicola Di Maio and his collaborators, I think he's in Nick Goldman's group, developed essentially a list of problematic sites, so positions of the reference genome that look to be frequently miscalled. And the way that they developed this list of problematic sites is looking for homoplasies in the phylogenetic tree. So recurrent mutations that look like they happened multiple times on the tree. So it didn't have a single branch that could explain the mutations. So this is presented as a VCF file that lists all of these mutations that may be erroneous. Now, we definitely use this masking file. Whenever I'm inspecting genomes that we sequence locally, if I see a site that may be problematic, we'll go and look up that site in this problematic site VCF file to see if other projects have had the same issue. Classic example is at position 11,083. There's a run of, I think, 9 T bases, and that position is very hard to amplify and sequence. So it typically accumulates both, I think, artifacts and also true mutations there, as it might have a little elevated mutation rate. So that file collects these sort of issues. We had a discussion in Cancogen whether we wanted to directly mask out any sites that appeared in this problematic sites VCF file. And we ultimately decided that we wouldn't mask the sites up front, but everybody's aware of it. So they can go and use that as a reference if they see any positions that they're uncomfortable with.
Yeah, and that's a great answer. A couple quick questions that we're kind of over time, but I think I'd much rather answer the questions. So Clement's asking, if the bound files are not generated by the ARTIC protocol, can he still use ncov-tools? I'm not sure if there's like a particular use case you're thinking of, because obviously the ARTIC pipeline should generate the BAM files, but do you know the answer to that one?
Yeah, I'm going to have to double check, but I think that the BAM files are a hard requirement where it won't run unless the .bam files are present. Both Richard and I are in the Slack channel, or the Slack for this course, so we'll double check that and get back to you, but I'm pretty confident that the BAM files are required.
Yeah. Kurt asked why anyone would sequences deep, I guess referring to the NovaSeq. I mean, I think that's just because for convenience, if you've got 384 barcodes and a NovaSeq sitting around, you might use it. I think it's not for any particular biological question, because sequencing extremely deep doesn't seem to add any information. You know, you're hitting into PCR and RT error way before you're finding biology if you're going to 100x, that 100,000x. So I don't think there's any good reason to do that other than convenience. Last question then from Ronnie. Have you tried these QC tools on wastewater samples?
I personally haven't, but I know some people have. I think John Tyson may have. Not all of the QC criteria that we use are going to apply to wastewater, but the basic things like the coverage plots, you know, the amplicon depth plots, those are all applicable and very useful for, you know, viewing wastewater data. But yeah, you would want to take some of their criteria with a grain of salt. Like obviously, you know, you'd expect more mixed positions if you're sequencing wastewater because it's more of, you know, a population sample. So you'd expect some ambiguity and some true mixtures there. So, you know, you might want to turn off the mixture checks, but as long as you can structure the data in the format that ncov-tools likes, it would probably run on wastewater.
Perfect.